Okay, so in this video we're going to continue our discussion on the vestibulocochlear nerve. In the last video, so let's write vestibulocochlear nerve. And what is this guy used for? Well, vestibulo is for vestibular system, cochlear is for cochlear system. What the hell does that mean? Vestibulo, eyes, right, for vision, balance, equilibrium, etc. Cochlear, ears, hearing. Okay, cool. So we're going to have the cochlear region, the cochlear branch, I should say, and then the vestibular branch, right? This nerve is divided in two. Okay, last video I spoke about the vestibular branch, so in this video, I'm only going to talk about the cochlear one. Okay, in the last video, if you guys remember, I had two different branches coming down, vestibular and cochlear, and we'll talk about that in a second. Okay, so now before I erase this and talk more about the video, I just wanted to mention that last video we spoke about the cranial nerve 8, right, vestibulocochlear nerve. Now we also only mentioned the vestibular branch. Okay, in this video I want to talk about the cochlear branch. Makes sense, because we have two different types. Now, cochlear branch, it's not that bad. I mean, I don't know. The vestibular branch is pretty straightforward. This isn't too shabby either. Just remember this picture in the last video we had. We had kind of like our inner ear. We had the semicircular canals. We had the utricle and the sacula. We had the cochlear, which we didn't speak about. But here, it's going to be kind of important. So what do I mean? So real quick, let's do the vestibular branch. We have semicircular canals, right? And then we have information coming from where? The ampulla, specifically from the semicircular ducts. We also have innervation coming from the utricle and the saccule via the macula, coming through where? They're meeting. Both of these things are joining together to form the vestibular branch. Now, I also said the cochlear, right? The cochlear receives information and it forms the cochlear branch. When these two nerves form together, they're going to form the vestibulocochlear nerve, cranial nerve 8. Cool. Now, the vestibulocochlear nerve, so vestibular branch and cochlear branch, are going to travel through the internal acoustic meatus. Now, also we know the cranial nerve 7, or cranial nerve 7, the facial nerve, travels through the, what? Internal acoustic meatus. Where is this guy located? He's kind of located outside by the temporal region, but his main goal, or where the fibers pass through, is the pons. Let me draw a little spinal cord right here, like that. And my brain, whatever. The pons medulla junction. So the fibers are going to pass right through here. We know that so the vestibular branch, right, he passes through the internal acoustic meatus and he passes through this same region, the pons medulla junction, and he's located where? He's kind of located in the, it's a really bad picture, I'm sorry. He's located in the medulla. Okay, so the vestibulonuclear complex is in the medulla. Now if I erase him, just to save space, the cochlear branch, he's going to pass through the internal acoustic meatus as well. He's going to end up in three different regions as well, mainly in the medulla region, superior region of the medulla, which is known which was known as what? The superior, the pons medulla junction is the superior region of the medulla. Good. Now, the facial nerve also processes through. We'll talk about it in a later video, but I just wanted to mention that real quick. You see here, I have it blown up. What do I have blown up? Okay. I'll erase this. Sad, I know. Done. Okay. What is blown up? Right here is our internal acoustic meatus. Here is the medulla, the pons, and the midbrain. So here's the pons and medulla junction. Here's our three nuclei that I said are going to be in the superior region of the medulla. What is these three things called when they're put together? These things are called the cochlear nuclei. Okay, so these are the cochlear nuclei, and we'll get into them more in depth. Now, I said the fibers come from the cochlear branch, right? This is the one we're going to focus on in this video, the cochlear branch of the cranial nerve 8, vestibulocochlear nerve. So, does it look like they just come right from the cochlea? No, Obviously not. I just drew that for that branch or that division just to make it more visual, like easier. Now I'll explain where these fibers really come from. Okay. Now, if we focus on the bottom half, right? This, I'll talk about them real quick again. But let's just focus on this right here, right? This is the ear. Okay, what part? Well, this is the auricle and the pinna, like our outside ear, right? And then we have a canal that travels through. What is that canal? It's the external acoustic meatus. Cool. Okay, the external acoustic meatus is then going to target 3 different bones in our inner ear. What are these 3 bones? We have the malleus, the incus, and the stapes. Okay, what do these guys do? I'll get to it right now. Okay, so the malleus, incus, and stapes, right? We're going to get, say we're listening, right? We're hearing. Something's going to come through, mainly air pressure is going to come through. It's going to target these bones. These bones are going to vibrate. When these bones vibrate, they're going to target this oval window. When they target the oval window is then going to target the cochlea. What's the cochlea responsible for? It's going to be fluid-filled vibrations. Okay. So say take this whole entire thing, right? Blow it up again. So we went from this, now to this. Blown up again. Okay. Now, here. This is the malleus, here's the incus, here's the stapes, right? These bones blown up. They're going to target this structure, right? In this structure, we have a couple different things. Up here, we have the oval window. Down below, we have the round window. Whatever. Now, we have 3 different regions as well. We have the scala vestibuli, we have the basilar membrane, also known as the scala media, and then we have the scala tympani. Okay, so we have VMT. Vestibuli, media, tympani. Cool. Now, the oval window, right? When the stapes vibrates, it's going to send a signal to the oval window. The oval window can then send a signal into the scala vestibuli. What signal is this going to be? It's going to be amplitude and frequency. Now, if we get a good frequency from this scala vestibuli, it's going to target the basilar membrane, also the scala media, also known as the cochlear duct. Okay, crazy. But they're going to send vibrations. So the amplitude and frequency is going to send certain vibrations. Now, why are these vibrations important? These vibrations help control a couple things. On the bottom here, you see I try to draw my hair cells. These hair cells are known as stereocilia. Whatever. Now, this red guy, he's like a muscle, right? This muscle is called a membrane, actually. He's called a tectorial membrane. If you can see, tectorial membrane. Cool. Now, on the tectorial membrane, you have something that comes off of him called kinocilium, or kinocilla. Now, when we get the amplitude and frequency, it sends to the vibrations, right? Depending on the vibration, the hair, so if here's a tympani, right? My arm, and the hair cells in my fingers, right? They're going to move. If they adjust like this, like little, I guess, tentacles, I guess you'd call it, right? If they adjust and they stimulate this tectorial membrane, something's going to occur. An action potential, well, the cell, I guess you could say, is going to be depolarized. And a depolarization causes what? We've said this, I don't know how many times. Depolarization releases neurotransmitters. What are the neurotransmitters that are going to be released? It's going to be glutamate. Now, glutamate is going to travel down, and when he travels down, he's going to come back into this region. He's going to proceed, and he's going to target the pseudo-unipolar neuron. This neuron can go 2 different ways. Okay, it's at the spiral ganglion. That's what it's called, the spiral ganglion. Okay, one way it could go is it could go to the hair cells, known as the peripheral process. So from the hair cells to the ganglion, known as the peripheral process. And it's really targeting the spinal organ of Corti. So this is the spinal organ of Corti. Whatever. Now, if it goes to the central nervous system, this is going to be our central process. So peripheral is telling our spiral ganglion to do something. What's it going to tell it to do? Okay, this spiral ganglion is going to tell the central nervous system, or the central process, to come down, travels down, and he forms, okay, there's a lot of these pseudo-unipolar spiral ganglions, right? Say there's 500,000 of them, right? They're all going to condense. When they all condense, they're going to form our cochlear branch of cranial nerve 3. We already said we have the vestibular branch and the facial branch going through the internal acoustic meatus. Now all we have to worry about is the cochlear branch, right, of the vestibulocochlear nerve, travels through the pons medullary junction, and what does he do? He's going to innervate 3 different nuclei, the cochlear nuclei. Okay, so we just spoke about the way the vestibulocochlear nerve and the facial nerve go through the internal acoustic meatus and travel into the, what is the structure? We've said this hundreds of times. This is the midbrain. This is the brainstem. What is the bottom region? Medulla, pons, midbrain. So this is a whole brainstem. Cool. Now, I know in the last video you didn't see all these writing and all these words because I couldn't fit it all on the board, but I'm going to get back to it. No problem. Here we go. So we said how this structure works, what occurs. Now, the cochlear branch, right, cranial nerve 8 in particular, travels through the internal acoustic meatus at what region? The pons medulla junction. At the pons medulla junction, it goes into 3 different colliculi, right? This is called the cochlear nuclei. Let me write it in real quick. I spelled that wrong, sorry. Cochlear nuclei. Okay, so from the cochlear nuclei, right? Cochlear nuclei got 3 different parts. It's broken up. We have a dorsal part and a ventral part, right? Anterior. Now, this anterior part is broken in half again. What are they? The dorsal part of the anterior part, I know it's a tongue twister, is the ventral posterior cochlear nucleus. Okay? And then the anterior part of the anterior part is the anterior ventral posterior cochlear nucleus. What does this even mean? Okay, so these 3 cochlear nuclei, right? I'm going to explain it again. So the red region, right? This is going to be dorsal. The dorsal what? I have it right on the board. This is the dorsal acoustic striae, right? Also known as the dorsal cochlear nucleus. I'll explain why it's a striae after. But it's a dorsal cochlear nucleus. Now, the royal blue one, right? This guy, he's going to be the ventral posterior cochlear nucleus. So I'm going to just draw him in royal blue, and I'm just going to abbreviate him. Ventral posterior cochlear nucleus. Okay, and then we're going to have the one in dark blue. It's the anterior ventral cochlear nucleus. Jesus. Why is this like this? Ventral anterior cochlear nucleus. Okay, now, this nuclei, right? This is known as the cochlear nuclei. I'm going to circle it. All 3 of these structures are the cochlear nuclei. They're divided. How are they divided? Okay, so the first region, right? It's divided into, not thirds, but into a 3rd part. So the top region, right, the most superior one, is going to be its own segment, the dorsal cochlear nucleus. Now, the inferior part is going to be this whole bottom region, right? It's going to be the ventral posterior cochlear nucleus and the ventral anterior cochlear nucleus. Now, this part is divided in half again. Okay? So, if we're looking at this structure, right, it's called the ventral posterior cochlear nucleus. Doesn't posterior usually mean behind? Yeah, it's behind the cochlear nucleus, but which part? It's behind this segment. So it's not all the way up here. It's not with the dorsal cochlear nucleus. He's the most posterior one. This guy's in the middle, that's why it's ventral, posterior. So in the ventral region, this is the ventral region, and this is the dorsal region. So it's in the ventral region, but it's posterior. This one is going to be the ventral anterior. So ventral region anterior, ventral region posterior, cochlear nucleus. These 3 guys are going to have 3 different functions, unfortunately. But that's mainly the whole thing we got to talk about in the video. So let's get back to it. Now, these 3 nuclei, right? They're going to have different functions. And what is this used for? Auditory. Okay. So first, let's go. We're going to start off with the red one, the dorsal acoustic striae fibers. Okay, the red one is the fibers, but the main thing is the dorsal cochlear nucleus. We just spoke about him. What is he going to do? So he's going to send his fibers, right? Let me get out the red marker. And these fibers are going to travel contralateral. So they're going to cross over. So I'm going to start over here. So it's going to cross over contralaterally. And where is he going to ascend up into? Well, they're going to ascend up and they're going to target this nucleus right here. We spoke about this nucleus a lot. He's known as the nucleus of lateral Luminiscus. Okay. Where is he located? Well, this is the pons region. So they're coming from the medulla into the pons. Good. Now, what is this even used for? Well, these dorsal striae fibers, the red ones, are going to travel up into this nucleus, and they're going to do two different things. One of these things are going to directly target that nucleus, and the second thing they're going to do, are they're going to travel upwards, right? They're going to travel upwards, and they're going to be involved in a tract. We're going to talk about the tract in a little bit. Just so you know, it's involved in both sides. Now, the next thing we're going to talk about is the blue one, the royal blue one, the ventral posterior cochlear nucleus. What is he used for? Okay, so this guy has little cells on him. These cells are called octopus cells, literally like the fish. These octopus cells travel contralaterally again. However, instead of contralaterally crossing in between these two olive green regions, you know the olive is what? The superior olivary nucleus. Instead of crossing in between them, they cross laterally to it. So not medial, but lateral. So he's going to cross lateral to this nucleus, and he's going to travel upwards, or ascend upwards, into what? Well, he's not going to target this lateral lemniscus. He's going to travel up, and he's going to form another tract. That other tract is going to target the inferior colliculus, which we spoke about, but I'll get to it again, don't worry. Now, the next one I want to talk about. Well, okay, so you know the red region, right? The dorsal cochlear nucleus, he has acoustic striae. Those fibers, the dorsal acoustic striae fibers. The blue region is going to have intermediate striae fibers. That makes sense. Dorsal is dorsal acoustic. Acoustic, so acoustic is for hearing. The middle one, so ventral posterior cochlear nucleus, is going to have intermediate acoustic striae, right? Striae is just the fibers. What do you think the anterior ventricular posterior cochlear nucleus is going to have? Okay, he's going to have ventral acoustic striae. That makes sense because he's the most ventral one. Ventral acoustic striae. Cool. What is he going to do? Well, he's going to do the same, basically the same thing as the intermediate striae. He's going to travel up contralaterally around the lateral side of the superior olivary nucleus, and he's going to ascend upwards. When he ascends upwards, he's going to be part of a tract. This tract we're going to get to, and he's going to target the inferior colliculus. Now, we know what our hearing, right? If we cover one ear, we're still able to hear normally, right? Our brain is not going to say, oh, only my left side of my brain could hear. We could hear on both sides of our brain. Why? Because we have bilateral crossing, right? Our hearing's bilateral. So these fibers not only go contralateral, they also go ipsilateral. So they're going to go ipsilateral, and they're going to target something. What are these guys? What are these guys going to target? They're going to target our superior olivary nucleus. Now, before I mention anything else, just know these three striae, right? These three striae travel upwards, and they're starting to form a tract. Now, I said the Luminiscus and the striae are also involved in this tract. Okay? How are these guys involved in this tract? I'll get to it right now. First, the lateral Luminiscus, correct? What does he do? Okay, so it's the nucleus of the lateral Luminiscus, and he accepts fibers from the dorsal acoustic striae. These fibers then ascend superiorly, right, into our inferior colliculus. Cool. Now, what's cool about these two fibers, they have it's called bilateral hearing, so we could hear on our right side and our left side. So they have something called crosstalk. That means they talk to each other. If the right one's damaged, the left one will work. If the left one's damaged, the right one will work. Mainly for which structure, though? Our dorsal acoustic striae. And what is that for? The dorsal cochlear nucleus. Whatever. Now, if we keep ascending upwards, right? I said the ventral acoustic striae targets these green guys, the olives. What are these olives going to do? Okay, so the superior olivary nucleus, right? It helps with two different things. The first thing it's going to do, it's going to help with localization of sound. Okay, so we can localize where sound is coming from. The second thing it's going to do, if our sound, or if the sound is very loud, right? Our ears start hurting. If our ears start hurting, these fibers are going to start to innervate or turn things on and turn things off. We'll get to there in a second. Just know, if the sound is too loud, the olivary nucleus, right, sends a bundle of cholinergic fibers that release acetylcholine, which are going to help depolarize certain muscles. Fine. Now, from our acoustic striae, or from our superior olivary nucleus, the fibers ascend, so I'm going to draw them ascending, upwards. They're ascending upwards into the inferior colliculus. And they're part of the tract that I've been holding off for so long. Why? Because we know what this tract is. This tract is known as the lateral Luminiscus. Oh my god. Okay, so this is the lateral Luminiscus nucleus. Well, the tract is called the lateral Luminiscus tract. Okay. I'm going to circle it. This bundle, right? It's the same thing on both sides. I just want to go on one so it doesn't get sloppy. This lateral Luminiscus pathway helps us with so many different things. What is it going to help us with? Okay. So all the striaes ascend up. What are the three different striaes in particular? Dorsal acoustic striae, intermediate striae, ventral acoustic striae. Fine. Now, when it goes to the inferior colliculus, right? This branching, the branching of the inferior colliculus also has bilateral connections. So they talk to each other. Okay. They're going to talk to each other. Crosstalk. Now, these fibers are going to travel up through something called the brachium. What type of brachium? Ow. The brachium of the inferior colliculus. What is cool about this brachium? These fibers we've spoke about so many times. What do these fibers even do? Well, they target the thalamus. Okay. Our thalamus was our main guy we spoke about in a video a while ago. He had 14 different nuclei on him. The main one we're going to talk about is the medial geniculi. Wait, why not the lateral geniculi? Well, the medial geniculi, I should have drawn him more medially, I'm sorry. This is the medial, nope. This is medial, lateral is going to be over here. So we're targeting the medial geniculi. And how are we going to remember it's medial geniculate and not lateral geniculate? Because they sound the same. Well, here's a little acronym or mnemonic, I guess, whatever you want to think of. We're going to say lateral geniculate nucleus is for vision. And how are we going to know it's for vision? Well, the letter L, right, stands for light in my mind. Light stands for what? Your eyes, like you could see, which is going to equate to vision. Cool. So lateral geniculate nucleus is going to be for vision. Also, one quick thing about the lateral geniculate nucleus. I know it's not in this video and it's not part of this topic, but what colliculus does he come from? So he comes from the superior colliculus. Okay. Where is this located? Okay, we spoke about the midbrain. The midbrain is what? The area above the pons. And the midbrain is divided into two segments. It's divided into an anterior colliculus segment and a superior colliculus segment. Okay, so the superior one is mainly for vision. So are we even looking at the superior colliculus? No. The inferior one is looking at what? Hearing. So we're only looking at the inferior one. I'll draw the supercolliculus up here just so you guys can see. Superior colliculus, and it would target the lateral lemniscus fibers. The lateral geniculate fibers, I apologize, but it's just side talk. Now, how do we remember the medial geniculate nucleus? Well, you could say it's easy. If the lateral one is L for light, the other one's got to be for hearing. Well, what if you forget that? Medial geniculate nucleus. Okay. M, right? M in my mind is going to stand for music. Music is going to stand for what? The ears. Ears are going to be what? Hearing. Okay, so you know this is for hearing. Now, what do these two things make up? The superior colliculus and inferior colliculus. When they're joined together, they're known as the tectum. The tectum is in the back of the brainstem, and the tectum is going to send different fibers, one for the eyes, one for the ears. We're worrying about the ones for the ears, remember that. Now, after this, he's going to send descending fibers through the descending tract to move the head and the neck. Oh, okay. So the inferior colliculus is going to send fibers. Let me draw them in, let's see, purple, I guess. He's going to send fibers down, right? Because this is like my brainstem. It's all the way up here. I'm going to send fibers down, descending fibers down, and I'm going to move the head and the neck. So I'll be able to, if someone is running, right, and I hear someone screaming, how can I determine, why do I want to look at them? Well, my ears are going to be like, oh, there's sound coming from this way, so I'm going to turn my head, turn my neck, then my eyes are going to move. What's going to tell my ears that the sound's on the left side? Inferior colliculus. from the medial geniculate nucleus of the thalamus. What's going to tell my eyes to move to the left? Superior colliculus from where? The lateral geniculate nucleus. Now, how does this happen? Or what does this do? What does this help with? Well, it's coming from mainly the superior olive. The superior olive is helping us localize where the sound is coming from. The sound comes from the superior olive, up into this inferior colliculus, into the medial geniculate nucleus, telling us where we're trying to move. Fine. Now, what else do I want to talk about this? This main inferior colliculus, right? He's responsible for the tectospinal tract. Wait, I just said the tectum. Yeah. So the tectum is going to have a tectospinal tract. This tract is going to involve fibers that go to the eyes and the ears. Right? Here's a little ear, I guess, and here's like a little eye. Little eyelashes. Cool. Now, what is the tectospinal tract responsible for in the inferior colliculus? It's going to be responsible for our auditory reflexes. So our reflexes, when it involves hearing, someone screaming, we don't even think about turning our head. Our head just turns. This is the reason why. We're almost done. We're almost done. Now, I'm going to erase the superior colliculus because he's irrelevant in this conversation, unfortunately. Okay, he travels up to the thalamus. Now, from the thalamus, right, we got to go a certain way. So we're done talking about the reflexes. We know the reflexes are quick. It doesn't even go up to our brain. We have memory involved in these tracts that automatically could do things for us. Now, say someone asks us a question and we want to respond to this question. How are we going to do this? Here we go. So, we're going to travel up into the medial geniculate nucleus, and then we're going to travel into the thalamus, and the thalamus is going to release different fibers. These fibers are going to travel laterally through what? They're going to travel through a structure. We'll talk about the structure in a little bit, but mainly it's called the transverse gyrus of Hesche, also known as the superior temporal gyrus. Okay, so why would it make sense for it to be the superior temporal gyrus? Well, it's going to be a gyrus up here. We know it's superior, and it's coming out of the temporal region of our body. That's where our ears are. Okay, that name makes sense. Why would transverse gyrus of Hesche make sense? Well, this is moving transversely. It's still a gyrus, and the guy Heschel is the one who found it, so that's how they named it that. It's kind of irrelevant, but I don't know, I like those little things. Now, these fibers that ascend out through these gyruses are known as auditory radiations of the temporal lobe. So they're going to the temporal lobe. Okay? We spoke about this. The occipital lobe in the most posterior region of my brain is known for what? Memory. More than just memory, obviously. It's known for vision as well. But just think about occipital lobe, and then it's got different regions. The hippocampus is back there, whatever. The frontal region of my head is known for reflexes, visualization, processing things. The parietal lobe. Now the temporal lobe, right? The temporal lobe is mainly known for hearing. Other functions as well. But we're going to travel into the temporal lobe. Okay, when we travel into the temporal lobe, what are we going to be targeting? Okay, so this is my really bad structure of the brain stem. Real quick. What is this region? Spinal cord. This. Medulla. Pons. Let me draw midbrain. Small midbrain. Cool. So, the first thing that's going to happen, these fibers, they're going to travel. They're going to travel out of the thalamus through the transverse gyrus of Hesche, right? I'm going to make a little canal. Bear with me. These fibers are going to travel through, and they're going to target this region of our brain. What's this region called? This region is called the superior temporal gyrus, which we just spoke about. Now, from this gyrus, right, he travels through where? The Wernicke's area. Or Wernicke's area, I apologize. Why is he traveling from this region to another region? Well, what is this guy even known as? He's known as the primary auditory cortex. Oh, okay, so if he's the primary auditory cortex, primary is our main guy, he makes us aware of sound and our ability to perceive it. So our body's oh, okay, I hear the sound and I can perceive the sound. What am I going to do with the sound? Well, I'm going to send it to Wernicke's area. And Wernicke's area is very important. Two main important areas for speaking and hearing. For hearing, it's Wernicke's area, comprehension of speech. For speaking is known as Broca's area, so they have to be connected somehow. How are they connected? Well, this region right here is called the Arcuate Fasciculus, and he's going to literally connect the Wernicke's area to Broca's area. Oh, so someone's like, hey buddy, what do you want for dinner? And you're like, you're not just going to say, what do I want for dinner? You're going to respond. Say like, I don't know, I want to go to Chick-fil-A. How am I going to respond through that? The Wernicke's area is going to send the fibers through the Arcuate Fasciculus, who's going to talk to all the different cortexes. What are the different cortexes? Prefrontal cortex, postcentral cortex, gyrus, etc. Those fibers are going to send descending fibers down into Broca's area. This Broca's area is in charge of our speech muscles. Okay. So if we're in charge of our speech muscles, we could hear the process, we could think about the process, and we could say something. Sometimes, like, if someone asks you a quick question, you could respond without even, like, thinking or processing. How? This tract right here. Okay. Now, a couple things I want to talk about. Before I talk about anything else, just remember, this is the only way, like, this is how our hearing works. It's really not too difficult. Remember, we have three different striae that travel up. They travel into the superior olivary nucleus, and then into the nucleus of the lateral meniscus, form the lateral meniscus tract, hit the inferior colliculus, travel up into the medial genicular gyruses, or luminiscus, proceed out through where? Auditory radiations of the temporal lobe, down the transverse gyrus of Heschel, or superior temporal gyrus, into Wernicke's area, through the Arcuate Fasciculus, out through the Broca's area. Okay. Now you're like, okay, well, that's cool. I understand how hearing works. What if you're at a concert and the music is so loud? So loud. Your ears are going to start hurting, right? You know how, like, on your phone it says, warning, like, the sound decibel is over a certain level, and you're going to damage your ear. What are you specifically going to damage? Here we go. So first, our main man, here, Olivary colliculus, right? Does two different things. I forgot to mention him before, but I'll mention him right now. The left side determines the intensity of the sound, so the lateral side, I should say. And the medial side, the relative timing of the sound. Okay, so an ambulance is, here's a perfect example. You're standing there, right? An ambulance is coming. All of a sudden you hear like a faint sound, right? And how do you know if it's from the left or the right side? Well, the superior olivary colliculus is going to help adjust to where it is, the intensity, and localize, right? So you hear the ambulance coming, you're like, oh, it's pretty far away. And it's on the left side. All of a sudden it's getting louder and louder and louder. So you're like, oh, it's coming towards me because it's getting louder. It's getting more intense. It's intensifying. The ambulance is right down Broadway, right in the middle. All of a sudden you hear it like on either side, but like you can't really distinguish it. Your right side starts to hear it and you're like, oh, this is so loud, so loud. All of a sudden it's starting to go further and further away from you. The sound's getting more dull. It's getting less intense. So you could figure out how far away something is based on the intensity of the sound. This is coming from the superior olivary nucleus. Now, say that ambulance is right next to you, right? And it's just ringing like an hour. Your ears are going to be like, yo, I'm going to go deaf. How are we going to do this? So one of the ways, there's two ways. One of the ways is going to be from the superior olivary nucleus. He's going to control something. The other way is going to be from our articular formation, which we'll talk about after. Well, let's do the superior olivary nucleus first, right? So he's going to get fibers. The fibers are going to come from where? The fibers are mainly going to come from our ventral acoustic striae that's going to it, or from our bilateral hearing, our crosstalk from our lateral meniscus nucleus. He's going to send fibers down and be like, yo, buddy, you got to do something. Like, I'm going to go deaf. Okay. Fibers come down, right? Fibers travel down, and where are these fibers going to travel to? These fibers are going to travel. I'm going to try to draw this as best as I can. Chondric fibers travel down, right? And what are they going to do? Okay, so they're going to release something called acetylcholine. Where is the acetylcholine going to be released? At the hair cells. So here's our hair cells, right? Can you see? Here's our hair cells, right? And it's going to release acetylcholine. Okay, so I'm going to blow it up a little bit. And what's it going to do? Okay, so if we release acetylcholine, we're going to hyperpolarize. Hyperpolarize. What does this mean? We spoke about this. Depolarizing is bringing something up to the resting membrane potential and hitting what? Plus maybe negative 35 from negative 70. Hyperpolarizing is bringing something down below the threshold around negative 90 millivolts. If you reach this threshold, something can't happen. What can't happen? Well, we can't contract. What can we not contract? We cannot contract this basilar membrane. Why would we want to contract the basilar membrane in normal terms? Well, we have pressed in molecules, right? These press the molecules. How are they stimulated? Air, right? We get a loud sound. To these three bones, what are these bones? The malleus, the incus, and the stapes. These bones, right, they push on this cochlear. They push the oval window and they try to hit the scapula or the scala vestibuli. These guys then push vibrations into where? The basilar membrane. So into the scala media or cochlear duct. When you have vibrations in this membrane, the hair, the cilia, remember we spoke about it. Here's the tectorial membrane, right? And here's the cilia. The hair follicles go like this. They stimulate that membrane that's like, oh, something's targeting it. This is how we get our hearing. Now, if we hyperpolarize this thing, right? This whole entire system, or muscle in particular, what muscle is this going to be? The muscles that connect right here, the stapes to the cochlear, can't stimulate. If they can't stimulate, we can't have this contraction. Our hair follicles, specifically the stereocilia, connecting to the kineocilium of the tectorial membrane, won't be able to work with hand in hand with each other. So it's going to stop our loud noise or hearing. It's going to be like, yo, something's not there, just turn it off. Now, these bones, right? We have a development when we're like old age called osteosclerosis, right? And it's aging. Osteosclerosis is going to then target these bones, degenerate them, and we're going to have something called conductive deafness. So the bones don't work as well, and we're going to start becoming deaf. Now, not last one, but another thing. We have something called articular formation, right? We've spoke about him a lot. I didn't really draw him in, but I'll draw him in now. He's right in the middle. Articular formation, and he's like, he tries to control a lot of different things for us. What is he going to do in this aspect? Well, he's going to target two different nuclei. Okay. He's going to target this guy over here and this guy over here. What are these going to do? What even are they? Well, the baby blue one is going to be our cranial nerve 5, and the pink one is going to be cranial nerve 7. What are the differences? Cranial nerve 5 is a trigeminal nerve. Cranial nerve 7 is a facial nerve. And how are they helping us with toning down sound? Because the articular formation is going to help us with loud sound. It's going to help us protect us. Cranial nerve 5 is going to travel down, right? We're going to travel into, let me see if you can see. Here's our ear region again, right? This is the auricle or the pinna, the outer ear. This is the external auditory meatus, the tube. Then we have the three bones, the malleus, incus, and stapes, right? Here's the oval window, here's the cochlea. Here's a super blown up visual. A less blown up visual. Here's a super blown up visual. Now, for the cranial nerve 5, the trigeminal nerve, right? He's going to stimulate a muscle called the tensor tympani. What is this guy going to do? Well, this guy, honestly, he starts the whole cascade effect. So he's going to target the malleus, right? And he's going to tell the incus to react, which is going to tell the stapes to react, which is going to react the oval window, which will then overact the cochlea. Now, when you interact with the cochlea, we're going to send fluid vibrations. These fluid vibrations travel through what? The cochlear nerve into internal acoustic meatus. However, if we target this tensor tympani, our main goal is we're going to contract it. So we're going to make this muscle really tight. What is this going to do? He's going to dampen the sound stimulus. So if this muscle isn't contracting, like it's like a drum, right? Like this. If this muscle isn't going boom, 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 and it's just like this, that sound can't ricochet. If the sound can't ricochet, we can't cause the vibrations, and we can't proceed through this cascade effect. Cranial nerve 7 does basically the same thing, except he targets the stapedius muscle, which connects the stapes to the oval window. Same thing. He pulls on the stapedius to help prevent it from acting like a hammer, like the doom, 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 doom. He just makes it tight, so it's just like stuck together. When that occurs, the rest of the cascade can occur. The fibers or the stimulus can travel through our Pons medullary junction, through the internal acoustic meatus canal, can travel up to different nuclei, can't go to our auditory radiations of the temporal lobe, can't go through Wernicke's area, Broca's area, etc. So it's helped dampening our auditory stimulus by literally shutting down the muscles that control the release of the vibrations and sensations, amplitude and intensity. That's it. There's one disorder that I want to talk about. It's called acoustic schwannoma. Okay, what is this guy? He's going to be a sensory neural deafness, right? You're going to have sensory neural deafness. And how? Okay, you could develop vertigo, which is nausea and vomiting, right? You think you're spinning all day. You could develop tinnitus, which is a ringing of the ear that doesn't go away. And you could have some hearing loss. And how does this occur? There's this different type of cell, or in our body we have cells, right? And there's a gene that's released. I'm not going to go into detail with the biochem, but that gene's going to release and it's going to keep multiplying, dividing, dividing, and dividing. What do you think occurs when cells uncontrollably grow and divide? Tumor. We develop a tumor. Okay. Where's the tumor going to be? It's going to be near the internal acoustic meatus or the cerebellar pontine angle. We know where the internal acoustic meatus is. It's right here. And then where's the cerebellar pontine angle? Probably in the pons, right? And then where's the cerebellar? Okay, well, we have the cerebellum. What? We have the peduncles. They all have peduncles. So here would be the intermediate cerebellar peduncles. This would be the inferior cerebellar peduncles. Here's the superior cerebellar peduncles. We're only talking about the intermediate ones, so the cerebellar pontine angle. Tumor, it's going to grow right over in this region, right? If it's growing in this region, growing, right? What nerve is it mainly hitting? It's mainly hitting this guy. This brown guy, right? What's the brown one called? The Cochlear Branch of the vestibulocochlear nerve. If that happens, we can send information into our pons medullary junction, and it can travel up through our brainstem. It's going to cause lots of problems. We just said them: vertigo, tinnitus, hearing loss, or tinnitus. I apologize. I know that was a lot. It got carried away, and I try to make it as neat as I can. But yeah, I hope this helped.